Perfect. So thank you. Appreciate the invitation to talk today. And I'm going to sort of change course here because we've heard a lot about mass spec and untargeted proteomics. And I'm going to hold up the torch for targeted proteomics, comparing a couple of different platforms that we had the chance to evaluate in our lab in the context of a COVID-19 study. So just to sort of get you acquainted with immunoassays that are using antibody-based detection or similar types of detection methods for targeted proteomics. You have, based on the number of targeted analytes, everything from single-plex ELISAs to very highly multiplexed platforms like Olink Explore and SomaLogic that use aptamers. But in the middle, you have this set of assays and platforms that are kind of 100 to maybe a couple of hundred analytes. And we find that to be sort of a cost-effective range. Maybe we can dream about doing many thousands of proteins, but if you have a whole lot of samples and a limited budget, maybe 100 or 200 is more reasonable. And if your question is limited, let's say, as is often the case for us, to inflammation and immunology, then maybe there aren't 10,000 things to look at, but maybe more like a few hundred. And so in that context, we tested this new platform from Alamar Biosciences called NULISA or NULISA-Seq for the multiplex version. And this is a proximity ligation assay, PLA. So for those of you who know the Olink platform, it's similar. Olink is a proximity extension assay. This one, both of them use pairs of antibodies that have oligonucleotide tags. And then those oligos either are ligated together as here, or in the case of Olink, are annealed and extended. In the case of the proximity ligation assay here, there are also two additional tags, one on each of the pair of antibodies that allow for additional capture and release steps, one with biotin avidin and the other with a poly-A tail that really cleans up the background. And so you'll see that is actually resulting in some increased sensitivity or detectability for low-level analytes. So I'll show you that data. But anyway, the readout is then by sequencing. And so you can multiplex a lot of these assays together and still easily quantitate the results pretty much without interference because of the high specificity of this PLA system and the sequential capture and release steps. And yet you can get good quantitation even for a couple of hundred analytes. There's also a fully automated instrument that goes with this system called Argo. It allows for up to 3 plates of samples to be run sequentially in a single run. Each one takes about 6 hours, and basically it gives you back a sequence-ready library. So that's kind of like a huge separator from other platforms that we have in the lab, where there's a lot of manual pipetting and manual wash steps. So this is the 250-plex inflammation panel that has been developed on this platform. As it stands now, I'm sure there will be additional versions, and there are new panels, including a CNS-based panel that's recently released. But this is the one that we were most interested in as an immunology lab. And we actually, in the data that I'll show you, used a slightly earlier version of this. It was like 203 initially, and then eventually the 250-plex. So how are we going to compare these? I'll show you in kind of 2 parts. One, more the technical comparisons between the 3 platforms I mentioned in the beginning, the Luminex, Olink, and NULISA. And we'll do that by looking at detectability in some COVID samples, ability to differentiate COVID from healthy controls in each assay, and then just also the correlations between the assays. And then in the second part, I'll show you a little bit more science in terms of what we could find in terms of predictive markers for COVID-19 severity and longitudinal changes over time. All right, so we published this work in a paper just a few months back. Koji Abe in our lab was the first author and really led this work together with folks at Alamar, including Joanne Beer, who did a lot of the statistics and analysis. And this is based on this large COVID-19 study called IMPACT that we participated in. We were a core lab for the Olink assay in this study, and we used actually the leftovers to do the comparison with Luminex and Neulisa. So I'm going to show you first in most of the technical data that I'm showing in the beginning, a sample set from this study that's very small. It's basically one plate, 23 COVID patients with various time points, mostly baseline, but some additional time points, and a range of severities, although most of them actually were in the severe category of 4. I'll show you what these trajectory groups mean in just a moment. But on this initial very small subsample, we looked, of course, at the ability to detect the common analytes between the 3 platforms in the COVID-19 patients. So each of these bars shows you the range of values in that subset of patients relative to the limit of detection for each assay. And I think this is a pretty telling way to look at it. You can see that there's just a couple of targets which are at or below the limit of detection for most of the samples in Neulisa. There's a bigger set for Olink that are at or near or below the limit of detection and of targets where the samples are below. And then for Luminex, there's also a somewhat bigger set, and there's just also more analytes that are closer to the limit of detection. It doesn't sort of rise as quickly, whereas with Olink, once you get past that initial set of poor performing analytes, the rest are really actually well separated from the limit of detection. So one thing that I should say about the Luminex platform with regard to this detectability question is that we do have a way to look at non-specific binding in Luminex. There's a company that sells these quality control beads called assay checks, and the 4th bead in their series, so-called checks 4, is actually a non-specific binding bead. And so we can use that to correct for the level of non-specific binding. And just to mention here that if you do that, you do improve the situation slightly with regard to analytes being at or below the limit of detection. So you can see that things raise up just a little bit on the right side versus on the left without the correction. So just another way to look at that detectability. Now here showing the percent of samples that that are above the LOD for each analyte, and there's labels on those analytes that are falling off below the 100% mark for each assay. And so you can see what I showed you in the other graph as well, that there's really just a couple for Alamar that are poorly detected. There's a bigger group for Olink, and with Luminex, you just sort of have this trend of things, not so many that are super poor, but you have a lot that are kind of not near, not really at 100%. And again, that's improved a little bit on the right when you do the non-specific binding correction. But still you have some falling off from good detectability. So I said we would compare COVID patients to healthy controls. And when you do that by the 3 assays, you find lots of things that are differentially expressed in COVID versus healthy. And I show this view just to sort of make the very obvious point that if you have 200 targets, you're going to find more things than if you have 92 or 80 targets. That's pretty obvious, but you know, it sort of says you need to look broadly if you want to find all the things that are different. If we instead then focus just on the 33 common targets between the three platforms, we still see that there are more. Now it's only slightly more, but there are slightly more targets that are upregulated based on the Neulisa assay platform versus Olink versus Luminex. Interestingly, there are more things shown here in Olink that are downregulated in COVID. And I'm sorry to say that I think that's an artifact that we introduced because the healthy samples for Olink we had to run on a separate plate because we didn't run them with the impact study itself. And so because there was a slightly different limit of detection in that plate versus the other, I think we produced this artifact. So I don't actually think that those are true downregulated targets. At any rate, we can also ask about the correlation between these assays. And given that Neulisa and Olink are similar in their detection method, they both have oligo-based pairs of antibodies, and one is sequencing and one is PCR, but they do tend to show the greatest correlation. So these are correlation coefficients for each particular target, and you see it as a histogram. So there's quite a few that are highly correlated targets, but there's also a kind of a drift off of some that are poorly correlated, and that probably has to do with different antibody pairs that the platforms use. We don't generally know from the manufacturers what the exact antibody pairs are. Now, if you compare either Neulisa to Luminex or Olink to Luminex, you actually get a worse picture. It's almost bimodal now, where there's a whole set of things that are really pretty poorly correlated. And again, that besides being due to different antibody pairs, could just be due to the very different nature of the assay in terms of Luminex being bead-based versus these other two. And here you just see those correlation coefficients shown in a bar chart form and identification of the poorly correlated analytes. And those often tend to be the analytes that are not well detected by one or another assay. So in the case of, for example, Alamar Neulisa versus Olink, some of these are poorly detected in the Olink assay and therefore not well correlated with Neulisa. Okay, last thing on the technical side, and now I will show you data from the larger cohort. This is 483 participants from the impact study. And while most of these are fairly well correlated, the dots fall fairly close to the diagonal, and you can at least see the R-squared values here, that some of them are very good. But there's a set of them for which there's some sort of leveling off of the correlation at a certain point, which suggests that we continue to get separation of or dynamic range in the Neulisa assay and not in the Olink assay. And yet there's yet a third category where the dots are really nowhere near the diagonal. They're just sort of spread along near the x-axis. And that's showing that there is dynamic range in Neulisa where there's very little in Olink. So I think it makes the point that while there's reasonably good correlation most of the time, there certainly are some instances where Neulisa is outperforming either based on dynamic range or just detectability, period. But all right, let's go to the next part where we can look at prediction and change over time within now this larger set of almost 500 samples from the impact study. And I mentioned about trajectory groups in the beginning. So this was something that came from one of the early papers from the impact consortium, was a way to sort of stratify rather than just sort of peak severity scores, looking over time at the kind of clinical scoring of patients. And I should say these were all hospital admitted patients. So they're all sort of severe in that sense, but you can see there's a group of them that sort of very quickly got better and were released. And that would be trajectory group 1. Trajectory trajectory group 2 and 3 are more moderate and took a little bit longer to recover. And then groups 4 and 5 had really sustained high clinical scores. And group 5, in fact, is the fatal group. So that's kind of the range from 1 to 5 in how they were scored. And even though we had almost 500 participants for the stuff that I'm going to show you here, well, I'll show you in one plot where we have all 5 groups labeled separately. But then for the statistical analyses, we ended up grouping together trajectory groups 1 through 3 versus 4 and 5, just because we thought that would give us a little bit better statistical power. All right, so this is still keeping them separate, and this is called a star plot, or specifically a penalized supervised star plot. It's something that our statistician Tyson Holmes invented, and there's a paper on it if you want to look it up in Journal of Immunology. It basically is a dimensionality reduction that is supervised in the sense that you tell the algorithm what the groups are that you want to try to see separated, and then it finds that plane through the data that best separates those groups. So here, each patient is labeled by their trajectory score. So there's 1s and 2s, 3s and 4s and 5s. And the vectors that you see are the major, not all, but the most prominent analytes that are basically separating the groups in this dimensionality reduction. So I think it's interesting because you can see that although there's some overlap, the 1, 2, 3, 4, and 5 severity groups are kind of arranged in a counterclockwise fashion. So there is some degree of separation, and the things that are sort of the vectors that are longest and that are labeled here are not even necessarily the targets that we think of with regards to inflammation in COVID-19. So I'll get back to things like IL-6 and TNF that people talked about since the beginning of the pandemic. They are differentially expressed with severity here, but they're not even the most major things that separate these groups. So the other thing that Tyson then did with these data from Neulisa is to do what's called an elastic net modeling, which is a way of doing multivariate modeling that kind of chooses a parsimonious set of analytes that best separate your groups. And again, we here for statistical power grouped 4 and 5, the severe groups together, and 1 through 3 was the comparator. So you can see all of these 6 proteins were picked because they were upregulated in the severe groups 4 and 5. And this was done using a bifurcation of the data set into a training and a test set, and the area under the curve when you do a plot of specificity versus sensitivity was reasonable when looking just at the test set after doing the training set. So I think it shows that you can, even with a relatively small number of proteins, separate the severity groups here. And again, maybe some of you recognize these analytes. I'm told that AREG has been in the literature for COVID-19 severity, but these are not necessarily the ones that I would have picked up as being popular proteins to look at for COVID severity. And again, I think it gets to the point that when you're looking at a couple hundred proteins, you are going to find new things that, if you just looked at the 10 things that everybody else looks at, you wouldn't find. So this is just taking the top hits from that elastic net model and showing you in terms of scatter graphs and with a logistic regression line how they perform in the test data. And yes, they are at least trending in the direction of being higher for groups 4 and 5. And then we can turn also to longitudinal differences and ask between the groups 1 through 3 and groups 4 and 5, are there cytokine targets that have a different longitudinal kind of pattern. And so in this test, there were lots of things that came up. It looks like, probably more than half of the targets in the assay that had some kind of longitudinal difference over time between severity groups. Most often I don't know if you can see, but most often when you have this difference, it tends to be that it's higher in groups 4 and 5 versus 1 through 3. So, upregulation of inflammatory cytokines in the most severe groups. But there definitely are different patterns here. We can focus in to see this a little bit better at just the ones with the most significant differences. And again, you see that it's generally higher in the severe group and often higher in the beginning and then begins to resolve at the later time points. And so maybe one more zoom in we can do. Oh, sorry, here is just highlighting CRP and IL-6. So coming back to the point that, yes, there are the usual suspects showing up as differentiated in this assay. CRP and IL-6, at least in the early time points, are definitely higher in the severe group. But we can zoom in one more time at just a few analytes that I thought were interesting in just the first 30 days. So I'm sorry, the scale here is no longer 500 days. It's the first 30 days only. Sorry for that. But here you can see some of these changes that happen really during the acute period of hospitalization. And I selected up at the top here the interferon alphas and interferon beta and gamma because those are talked about a lot. And we know that interferon alpha is upregulated in cases that are resolving COVID-19 well. And indeed, it's both of these are examples that kind of buck the trend of that I was saying a moment ago, where these are actually higher in the mild groups, at least at the beginning, and then it sort of equalizes or even shifts place in the later time points. But early on, that early production of interferon alpha is higher in groups 1 through 3. And a little bit less obvious for what's happening with interferon beta and gamma. It seems like they're only differentiating after the first couple of visits. Similarly, IL-10 seems to be higher, at least after a few days in the severe group, same with TNF. And IL-5 I threw in here just because I hadn't really heard about IL-5 in the context of COVID. And it had a very interesting pattern here where it seems the groups seem to cross so that the severe group is showing elevated IL-5 only after the first couple of visits. So that may be something related to eosinophil activation or something in the severe group that's happening over time. Okay, so with that, I would like to leave you with the idea that this NULYSA assay can successfully look at quite a few targets together and with excellent detectability. There are more samples below the LOD for Olink and Luminex than with NULYSA. And there's reasonable correlation, but better correlation with NULYSA and Olink, poor with Luminex and the other two. And to some extent, there's more dynamic range with NULYSA and for the good targets also with Olink. And that we can look at combination of targets to really predict, and it doesn't have to be that many targets, to very well differentiate the severe from mild COVID cases in this impact study. And that lots of different targets are longitudinally changing in COVID patients, usually with the higher severity ones having higher levels for pro-inflammatory targets at least, and some other interesting cases like the IL-5 I showed you. So I'll stop there and want to again thank folks in our lab that contributed, and as well as Alamar, and of course the Impact Network. These are some of the leaders, but those are the impact sites over there, and there's really like a few hundred people involved in that large study, and really was a great effort to be involved with. So thanks for your attention, and I'm happy to take questions.